Hello, good afternoon, everyone. Let's start by finishing the class poll questions from lecture 2. I think there were quite a few of them. Let me put them on the screen for you. And we're just going to go through the answers before moving on to today's lesson. So here, this is slide number 84. On the PowerPoint deck. Oops, 84 right here. Okay, so it says here a peptide bond forms between which group? So just as quick review for you, the amino acid has a central carbon that's connected to a hydrogen, and then we have a COOH on one side and NH on the other. And that's going to be what we call a side chain, right? Side chain. You have all these already from last class. Now this group over here, that is called a carboxyl group. Whereas over here, that is called an amino group. So when you have two amino acids side by side, the carboxyl group will join with the amino group to form a peptide bond. Okay, so the answer is C. If you think about the amino acid in the primary structure, right? What's linking them together would be the peptide bond. Okay, peptide bond. Next, here it says hemoglobin is an important protein that is composed of 4 polypeptide chains arranged in 3D shape. What level of protein structure is illustrated by hemoglobin's 4 polypeptide chain? Okay, so 4 polypeptide chains means you have multiple subunits, right, to come together to form a functioning complex, and that is what we call the quaternary structure. Okay, quaternary structure. So like you might have a red polypeptide, 2 red polypeptide, right? And then you will have 2 blue polypeptide. Right? So all these things come together to form the hemoglobin. Okay, and these are individual polypeptide. Okay, so as I told you in class, not every protein is going to have 4 levels. Proteins, some of them just stop after the tertiary, right? Some proteins are capable of doing its job when it's just by themselves. It's only certain kind of protein that requires multiple polypeptides to join and become a functional unit. It is in those cases where we would have to describe the quaternary structure of the protein, right? Which polypeptides do we need to put together, right? Like in this case, okay, 2 of the red ones and 2 of the blue ones, right? So that's that. Feel free to jump in if you have any question. Just ask in the chat or unmute. Next question. Pancreatic cells make insulin. If you don't know what pancreatic cells are, and if you don't know what insulins are, it doesn't matter. That's not important in this question. What's important is that you realize it is a type of protein. Okay. So these cells, as in the pancreatic cell, we'll use something to synthesize insulin. Synthesize is to make, right? So what do we need to make proteins? Well, you should have a summary chart that writes down the monomers for all the macromolecules. And in the case of proteins, we need amino acids. And we would join them together using dehydration synthesis. Okay. I will give you a moment to read through this one, and I will launch the poll for you to try. Okay, that was about a minute. I only have 4 responses though. Does anyone else want to choose something before I close the poll? I would recommend trying to, keep it under 2 minutes per question on the quiz, so you don't run out of time. Okay, so let's see. We're looking for the false statement. First one. Primary structure of a protein is determined by the sequence of amino acid. That is correct. That is precisely what primary structure is. You read the order of the amino acid that is in the chain. That is your primary structure. Example of secondary structure includes alpha helix and beta sheets. So remember these little spirals and the zigzag, that's the primary and secondary structure, right? So that is also a correct statement. We're not going to choose that. Tertiary structures are stabilized by hydrogen bonds of peptide backbone. Now, if you read the notes that we wrote last time, that is not true. Okay, the tertiary structure, which is the overall 3-dimensional shape, that is stabilized by interaction of the side chains. Okay, so the thing that is stabilized by hydrogen bonds of the backbone, that would be the secondary structure. Okay. Now, I also told you last class, if you change the temperature or if you change the pH or the type of solution, what you're going to end up doing is you will be disrupting the secondary and tertiary structure of the proteins, causing it to unfold, right? Causing it to denature. But the primary structure will remain intact. The peptide bonds are quite strong compared to some of the interactions that are holding the secondary and tertiary structure together. So if you crank up the temperature, only secondary and tertiary will be affected, but not the primary. So that is also a correct statement there. Okay. All right, and then we did all these stuff, and here is did we do? Oh yeah, we did these two already. Okay, so let's move on to this one right here. Okay, which of the following statements is true regarding purines? Okay, so purines include the A and the G. A and the G. The phrase I tell you to remember was pure as gold, right? A and G. And then the pyrimidine would be everything else. So the C and the T and U, but U is RNA only, right? And T would be DNA only. So let's take a look and see which one is true. First one, adenine's bonding to thymine is stronger than guanine's bonding to cytosine. That is not true. Okay. If you take a look at the picture here, you can see between A's and T's, there are only 2 bonds, 2 hydrogen bonds holding them together. But between C and G, there are 3 hydrogen bonds. Right. What that means is if you have a DNA strand and there are a lot of G's and C's in it, it's much harder for you to pull them apart. And that's actually an important thing to realize when we do some of the later units. Okay, so there are 3 think of them as like tapes, right? 3 pieces of tapes holding the C and G's together, but only 2 for A and T. So C and G's are much harder to pull apart than A's and T's. For that reason, the first one is not correct. It's the other way around. Next one, cytosine is a purine. Well, if I check my purine list here, there's no C, there's no cytosine. That is incorrect. Purines bind readily to deoxyribose, but not to ribose. So if we take a look at the purines, we have A and G together, but these are common in both DNA and RNA, right? Of course, deoxyribose is DNA and ribose is RNA. So basically what this is saying is you will find A and G in DNA, but not RNA. But that simply is not true, right? You will find it in both cases. Okay, next one, both adenine and thymines are purine. That is not true. Thymine is not purine. It should be guanine. And purines have a double-ringed structure. That's correct. Okay, if I go back here to show you the purines, right, you see purines have 2 rings. Okay. So some of the questions on the quiz will be like these, where you have to do quite a bit of thinking. Other ones will be relatively straightforward. Okay. Do we have any questions?
Is the quiz going to be today or is going to be the next time we have
The quiz is going to be available on Wednesday between 6 a.m. to 11:59 p.m. And we will have class as well during our regular time on Wednesday. Okay, so I actually post the information online for you guys. I don't know if you had a chance to look at that. I posted last class. All right, so Wednesday 24th, and that's going to be the link to your quiz. Make sure you do the scientific method self-quiz to familiarize yourself with how to do assessment online. This will also help you install the lockdown browser, which once again is going to basically prevent you from accessing anything on your computer while you take the test. That includes going onto the internet. So, you know, if you want to use the notes that we have in class, please print it out or, you know, copy it down, write it out, and have a summary sheet. That is okay with me. There will be 17 multiple choice questions, 5 multi-select questions. These are questions that have multiple correct answers. And then there will also be 2 short answer questions. Okay, the short answer questions are pretty straightforward. I will give you a scenario and you will have to tell me, you know, what the independent and dependent variables are. Maybe you have to write a hypothesis. And then the other one is I'll give you a picture and ask you to describe it using qualitative or quantitative observation. Okay, so those are the short answer questions. Sabrina, do you have a question? I see that you're mute.
Yeah.
Mhm.
Sorry, I'm new over here. So where I can get like summary notes for upcoming tests, like
Okay,
sample operations to
so
practice.
yeah, so if you go to the online eCentennial, the lecture archive is where you can find the previous recording for the class that you've missed.
Mm-hmm.
Class notes is the notes that we took in class in addition to the PowerPoint, and
Mm-hmm.
study guide answer. Okay, that's where you can find some of the, study questions. All right.
Sorry, like for the upcoming tests, if I study from this study guide answers, I'll get a question from there. It's related to this. Yeah.
Not all of it is going to be related to it, but then it will help, serve
Yeah,
as a
like
starting
I
point.
just I just try to look all the slide and it's kind of new, so
Mhm.
I need to practice for the exam. So that
Yeah.
is
So
want to
you
say.
use the PowerPoints for sure, and then use the study guides. Those would be the main materials that you have to use to prepare for the quiz. Okay.
Okay, thank you.
So time limit is 45 minutes, and like I said, it's going to be covering lecture 1 as well as lecture 2. Does anyone else have other questions related to the quiz? Is my quiz the first quiz, or have you guys done another assessment in another class already?
I've done one already.
Yeah, we did some quizzes and assignments.
You guys did some quizzes and assignment, okay? So like you should be familiar with how this goes, right? Okay. The main difference, I suppose, is like there's a big window. Oh, I guess I have to remind you guys, once you start the quiz, the timer does not stop, right? Like even if you turn off your computer or whatever, like close the browser and then you come back later, the timer is not gonna stop. So make sure you are in a quiet place where you won't be interrupted while you're doing the test. Okay?
Sorry, like other quiz we have just faced, they have like you can try twice, so it
So
will
only
be
one
one
time, only one
one
attempt. Yeah,
time.
only one attempt for this quiz. Okay?
Okay, thank you.
Yeah. All right, let's continue with some of these questions. Next one, which of the following is not a feature of DNA? So this would be an example of a multi-select question, right? Where there are more than one correct answers. And you know it's a multi-select question because the question on the quiz will tell you to select all correct answers. Okay. So first one, nucleotides within a single strand are connected by covalent bonds. That is true. Okay, that is true. But we're looking for the ones that is not a feature of DNA. Okay, anything that you connect using dehydration synthesis, it's gonna be a covalent bond. Okay, so when you connect two glucose together, that's a covalent bond. When you connect two amino acids together, that's a covalent bond. But sometimes we give them special names, right? For example, in the protein, that particular covalent bond, we call it a peptide bond, right? Okay, next one. Adenine forms base pair with uracil. That is not correct. That's the one that we're choosing, okay? There are no uracil in DNA. Uracils are only found in thymine, in RNA. In DNA, we have thymine instead. Complementary strands are held together by hydrogen bonding between phosphate group. That is incorrect. Okay, that's not true. The hydrogen bond are holding together the nitrogen base, right? Okay, the A's and the T's and the C's and the G's. That's what's holding the two strands together. complementary strands are anti-parallel, running in opposite directions. That's true. Right? I told you the two strands are like two sides of the highway, right? They are parallel, but one is northbound, the other is southbound, anti-parallel, opposite direction. Okay, this is the last one. And this would be an example of an application questions. Okay, something that is much harder and it requires you to process what it is that you've learned and apply it in a new situation. So we'll do it together. A chemical reaction occurs slowly at 37 degrees Celsius when molecule X is added. The reaction speed up significantly. Which of the following is a monomer of molecule X? So what is molecule X? Does anybody know? What did we learn in this lecture? Something that can be added to speed up reaction. Sorry, what was it, Fatima?
Enzyme.
That's right. So this is an enzyme. Very good. Enzyme speeds up chemical reaction, right? And you should also know enzyme is an example of a protein. So what the question really is asking you is to find the monomer of a protein. Okay, so what is the monomer of a protein?
Amino acid.
That's right, amino acid. So which picture here represents an amino acid? Well, that's right, option A. Right? That is an amino acid. We've drawn a couple of times, right? Now, just for your record, B over here, that is a nucleotide. You can see there's the phosphate, the sugar, and the nitrogen base, right? This here is a monosaccharide. And anybody know what this long thing is?
Fatty acid.
That's right, this is a fatty acid. All right, so that is all. Now, the questions that we just went through, these are considered as like the harder ones. Okay, it's not all going to be like that on the quiz. The quiz have straightforward questions where you should be able to find the answer relatively quickly. But then there are a few of these ones that you have to be a little bit, more careful of. And some of these questions will show up on the quiz as well. All right, so don't think just because we did it in the concept check, there's no way he's gonna put the same question on. Sometimes I will. All right, so please make sure you understand them and take the time to go through them before doing the quiz. That's it for lecture 2. Are there any questions from the study guide? Any questions, even from lecture 1 related to the upcoming quiz before I start lesson 3? Just gonna give you guys a moment to think about that. Okay, so again, the previous recording, some of you joined the class late. If you need the previous recording, you log on to the course homepage. If you don't know where the course homepage is, I will post it in the chat box. And you click on the lecture archive. That's all the classes that you have missed so far. Okay, this is our 5th class today, right? So everything here. Right. Anything else that I can help you with? Okay. Well, if you guys think of anything else, feel free to just, leave a message in the chat box. All right. So we're gonna move on to lecture 3 today, which is about cells, right? Which is about cells. So let's start with a basic introduction of what a cell is and what it does, and some properties of cells. Now, I just want to remind you that we are basically moving up this level of organization, right? We first saw this hierarchy in the first lecture, where we talk about characteristics of living things, right? Yes, go ahead, Lika.
Excuse me, Professor. Lecture 3 will be in the quiz or no?
No,
Just
no,
lecture
no.
1 and 2?
That's right. Quiz 1 is only
Okay.
lecture 1 and 2. Okay.
Professor, it means for the lecture 2 from the slides. So we will just read until lipid.
All of
Yes?
it. No, the quizzes include all of the
All the
lecture.
Macromolecules, all the
Yeah,
macromolecules.
all the macromolecules, yes.
Okay, got it. Thank you,
Yeah.
Professor.
No problem. So, this is the organization of living things, right? Which is characteristic of living things, right? They all have some kind of complexity associated with them, and the complexity and organization not just applies to the whole organism, but on a molecular level as well. Okay, so much of lecture 2 involves about learning, the atoms, right, that makes up a majority of a living organism. And then we learn about some important molecules, the small ones like glucose and amino acids. And we did the macromolecules, right? The carbohydrates, the proteins, the lipids, as well as the nucleic acid, right? So now we're going to move up the hierarchy. We're going to take a closer look at cells and the different things that makes up a cell. Okay? And we're going to... Yeah, go ahead.
Organelle something as organs?
Say that one more time, please. Sorry.
The organelle, this organelle here after macromolecule. Yes, is this the same thing as organs or as
No. So organ would be like, like a stomach, right? Like a liver or heart. That's an organ. Yeah. As you will see, organelle, the nail means like little things. Okay. So little organ. Yeah.
Oh, okay.
Yeah. So these organelles are going to be, as you will see, different parts within the cell that have designated function. Okay. So we're going to spend the rest of the course pretty much all the way until lecture 9 to talk about all the things that are happening within the cell. And then in the last lecture, we will move on to tissue. So we're going to stay in this part of the hierarchy for quite some time. Okay. Just giving you a heads up of what to expect. All right, so how big is a cell? Well, actually, a cell is pretty small. If you think about an adult, right, it's over a meter tall, maybe 1.5-ish meters. And then if you go down by magnitudes of 10, right, if you keep on dividing and dividing by 10, eventually you will get to this range, which is measured in micrometer. Okay. So 1 micrometer is 10 to the -6 meter. It's a millionth of a meter. Okay, so you take a meter and then you divide it by 1,000,000 times, then you get 1 micrometer. So animal cells and plant cells, they are measured in the range of 10 to 100 micrometer, depending on what kind of cells you're looking at. Some cells are bigger, others are smaller, depending on what kind of cell you're looking at and what their functions are. Some cells are very long, okay, like neurons, which is a type of cells that are found in the brain, for example. They could be quite long. Right? Cells like muscle cells, they're quite long. Others, they are much, much shorter. Okay. And inside the animal cells and the plant cells, you will have organelles, like I said, that will help them perform function. Those are just a little bit over 1 micrometer. And if we keep on going smaller and smaller, things like viruses, they are measured in nanometers. Okay, nanometers. 1 nanometer is a billionth of a meter. And you take a meter and you divide it by a billion, 1 with 9 zeros, then you get nanometer, right? And then on the lower range of the nanometer scale, you would have the proteins and the lipids and the the carbohydrates, the macromolecules essentially. Okay, so you don't have to memorize any of this. This is just to show you, on a scale how big things are. So these things are like pretty small. We cannot see it with our naked eye. That's why we need to use light microscope. Okay, and anything that's like even smaller, it get cut off here. All these things you would need to use a electron microscope to see it. So more on that later. Here is a slide that shows you the different types of cells and the variety of cells. In terms of what cells look like, they are as diverse as living things in general. So over here, this is a bacteria. Right? We call these coccus, right? Because they are like little sphere. Sometimes they grow in clusters. Sometimes they grow in chains. You might have heard of Staphylococcus, right? The one that caused strep throat. That is, that is a type of spherical bacteria. This over here is amoeba, okay, which is they live in like fresh water. Sometimes they make their way into swimming pools, and then they give people diarrhea if you happen to get them into your body. These are neurons. Right? Neurons. These are again coccus, C-O-C-C-I, cocci for plural. It's a type of bacteria. So even though the cells look very different, but fundamentally they are the smallest unit that has the properties of life. Now we learned about the properties of life in the first lesson, right? That they have organization, there is requirement of energy, that they are able to reproduce, right? They are able to maintain homeostasis. So all the things that we talked about will still exist on a single cell level. Okay? So that's what's defined as the cell. It's the smallest unit that still retains all these properties. Now, a protein, even though it's found in a living organism, it's not living, right? Because it doesn't have those properties. It's not able to, maintain homeostasis. It's not able to reproduce and so on and so forth, right? So a cell is the absolute smallest that you can go and still have all these properties. Now, even though the cells look very different, they all have common features. Here are 3 common features of all cells. They all have a cell membrane, which is also known as a plasma membrane. Okay? So like if you think of a cell, that's what this is. This is a cell membrane. Whether you are an animal cell, a plant cell, a fungal cell, bacterial cell, doesn't matter. If it's a cell, it's going to have a cell membrane. Does anybody remember the key component of the cell membrane? We learned that last week.
Phospholipid?
That's right, the phospholipid, right? Which is a type of lipids, the one with the head and the 2 tails, right? So that's the cell membrane. In addition, a cell also has genetic material. Okay, so these genetic material can be DNA, and sometimes it can be RNA as well. And finally, the inside of the cell has kind of this liquid gel-like material where everything is floating in. Okay? And that is what we call the cytoplasm. Okay? So those are the 3 common features of all cells. Even though they all look very different, different shape, different size, they will always have these 3 things. Okay? So there is something called the cell theory. Okay? There are 4 parts to the cell theory, and I'm just going to go through them with you. And the first part of the cell theory states that every living organism consists of 1 or more cells. Okay? So sometimes the organism is just 1 cell big, like this thing called the paramecium. Okay? It's same as the amoeba. They live in like fresh pond waters. Okay, and this is one cell, big only. We call this unicellular. Okay, unicellular. Some organisms are they are small, they are microscopic. You cannot see it with your naked eye. You have to use a microscope to see them, but they have thousands of cells. So this is called the C. elegans. It's one of the model organism. Model organism for research. Okay. So scientists knows a lot about model organisms. So this C. elegans is one of them. Okay, fruit flies is one of them, and then mouse as well, right? So we try to do experiments using these model organisms before we move on to actual humans in some cases. So these things are about 3,000 cells big. So one of the interesting thing I recently read is they found a gene, which is a segment of DNA, that they were able to alter in these C. elegans, which is a type of worm, okay? And they were able to extend their life by as much as twice as long. So that is research into anti-aging procedures, right, I suppose. So if you look at a human has trillions of cells, okay? Lots and lots and lots of cells compared to something like a paramecium, okay? But regardless how many cells you have, if it's a living organism, you must have at least one cells, and then there's no, upper limit. The bigger the organism, the more cells is going to be made up of. That's the first part. The second part of the cell theory states that the cell is the smallest unit of a cell, of life, sorry, smallest unit of life, right? Which I kind of explained to you already, right? It has all the properties of the living thing, and they are individually alive, even though they are part of a multicellular organism. So, we as a whole human being, we have our own thoughts and we can control our movement, but on the cellular level, each individual cell, they are alive on their own. In fact, you can take cells from the body and you can grow them in a petri dish. That's what this picture is. Okay, that's a petri dish. And the red stuff that you see there, those are like nutrients that you can feed to the cells to keep them alive outside of your body. Okay, of course, you cannot do that forever. Eventually, the cell will die when they're out of your body, but you'd be surprised. You can keep them alive for quite some time. Okay? They are quite happy to be not part of our body. So there is something called the HeLa cells. HeLa cells, that's what they look like under the microscope. I mean, they are black and white, but then like you can stain them with fluorescent dye and make them colorful when you look at them under the microscope. Okay, and HeLa cells is a very commonly used cell line in research. Okay, and it's the first cell line that has been used, that has been cloned and grown endlessly in the laboratory. So it was actually taken from this lady. Okay, her name was Henrietta Lacks. Okay, that's what the HeLa stands for. Henrietta is the first two letters of her first name, and then Lacks, that's the first two letters of her last name. And the person had cervical cancer, okay, at the time. And what happens is the doctor took a biopsy, a little sample from her cervical cancer tumor, and they were examining the cells under the microscope. And then what happens is they found out the cells were able to continue to divide in the petri dish. As long as you give them nutrients, they're able to divide and divide and divide. Okay, and so they keep on growing this thing, right? And the use of HeLa cells has been very, very important. in discovering things like new vaccines against like polio, right? Or coming up with treatments for like TB, Ebola, HIV, and cancer treatment. All these kind of research has used HeLa cells in one way or the other. So you can buy HeLa cells online, and they're not cheap. Okay, so like I just searched it up last year, end of last year, and you can buy. You can depend how much you want, how much you want to buy, right? Okay, so for 20 microliter, okay, so we already know micrometer, right? Micrometer is a millionth of a meter, right? So 20 microliter, 1 microliter is a millionth of a liter, right? So for so small amount, it's already like $486, right? So apparently the company that sold these, why people buy it? Okay, someone asked me why do people buy it? People buy it to do research, right? Okay, so like people who are doing cancer research, they have to first prove that it works on a cellular level before you move on to animals, right? Like mouse, for example. And then if it works in the animal, then you move on to the people, right? So you always use cells as a starting point. So that's why people buy it. Like you and I are not gonna buy it, but researchers, scientists, they're gonna buy it. So the people who took the cells from the patient did not actually got her consent to grow her cells. Okay, and the company that sells these cells, the Thermo Fisher right here, they essentially made a lot of money by commercializing the cell lines that were taken to the lady. So the woman died of the cancer eventually, but her family went into a lawsuit in 2021 against the company, saying that they didn't have the consent to commercialize the cell line, and that they are seeking for compensation. So the company tried to file their own lawsuit to dismiss the case, and then finally, right, in 2023, last year, they reached settlement. Okay, so basically the family got compensated, but then the company didn't admit any wrongdoing, right? That's how usually these things go. But that shows the importance of obtaining ongoing consent if you're gonna be growing cells that are taken from people. Okay, so this I guess the point is to show you cells can grow outside of the body, and if it's cancer cell, then they can grow endlessly. Okay, and if you're wondering how is it possible for cancer cells to grow forever, that is something we will talk about in lecture 7, I believe. Okay, moving on. We are talking about the cell theory, part 1. Living things are made up of 1 or more cells. Part 2, they are the smallest unit of life. Yeah.
Professor, you said cell can grow outside of our body, but I didn't get the second one. You said cell.
Yeah,
If
I said
that's cancer cell.
if it's cancer cell, then it can grow forever. Okay, theoretically. Okay, so as long as you keep on giving them nutrients, they are able to grow. Okay, if it's like a regular cell, if it takes some cheek cells and then grow them, even if you give them the best nutrients, they are not going to be able to last, more than a week or 2 in some cases.
Okay, thank you.
Okay, so the third part of the cell theory states that all living cells come from division of pre-existing cells. Okay, no one is 100% sure where the first cell ever came from, but to get new cells, you need to divide existing cells. So this picture here shows onion cells, and this cell right here is dividing. Okay, so that's one cell. And then if it divides, then it becomes 2. Okay. So we'll learn about the division process again in lecture 7 or 6, one of those. Last part of the cell theory, cells contain hereditary material. Right? These are the genetic material, right? The DNA and the RNA. And these genetic material are gonna get passed on to the next cell when they divide. So 4 parts of the cell theory. Now, to study cells, we have to use microscope. Okay, it's very small. The cells, but there's no way we can see it with just our eyes. And the first microscope was invented by a Dutch scientist by the name of Antony van Leeuwenhoek. Okay, so Leeuwenhoek built a handheld microscope like the one shown in the picture, and when he put some pond water under his microscope, he saw a whole world that he never knew existed and nobody knew existed. Okay, like if you look at a drop of water from the pond, right, like you wouldn't expect anything living in there, right? But you know, if you look at it under the microscope, you'd be surprised to find a lot of things, right? So he described these things as animalcules, okay, as in like little molecules that looks like animals, okay? But we now understand those are unicellular organisms, right? So this is called a basic light microscope. Okay, we have a lot of these at school. If you take this course in person, we would be learning how to use these microscope. But for online, we'll just be doing a simulation. Light microscope are relatively cheap. The ones we got from school, it's like about $300 each. So it's affordable for a classroom. But to see very small ones, like really small structures, the light microscope is not going to be enough. Okay, we have these things called transmission electron microscope (TEM) or scanning electron microscope. These microscope are really big. You can see it takes up like almost a whole room. And they are able to magnify up to 2.5 million times. Okay, whereas the light microscope, the ones I have at school, they could magnify up to 400 times. That's the maximum. Okay, 400 times. So at 400 times, you can see like your blood cells. Okay, but at like 2.5 million times magnification, you can see very detailed structure, even those that are within the cell. Okay, so these microscopes are really expensive. They could cost anywhere between $500,000 to $1 million. And you know, hospitals sometimes use them for diagnosis. Sometimes you know, lab use it for research, but it's not something that you know is commonly used in a school setting. All right, so these things that you're looking at, we'll learn later on. These are called cilia. They are little hair that are found in the airway. It helps you remove trapped dust particles, right? All right, I have been talking a lot. Let's see if you learn anything from me. Okay, so please try to participate in the polls. Okay, thank you. So which of the following is a concept of the cell theory? Most of you got it right. It is D. Okay, it's not A. Two people choose A. It says organelles are basic unit of life. Nope, cells are basic unit of life. Okay? Organelles are the structure that you will find inside a cell. And we'll learn more about those for the rest of the class. Okay, here's another one. Okay, all of the following are present in all cells except, so if we go back to our previous slide here, you can see that we've drawn this, right? Common features of cells include cell membrane, also known as plasma membrane, genetic material, and the cytoplasm. So if you compare that with the list here, that's in the cell, that's in the cell, that's in the cell, right? So even though you might not know what this is yet, but that's not on the list. That's why this is the exception, and that's the answer. Okay. So that's an overview of cells in general. Let's take a closer look at the cell membrane as well as the nucleus. Okay, but before that, we should compare two kinds of cells. We can categorize a cell as eukaryotic cells or prokaryotic cells. Okay, so eukaryotic cells, which are also known as eukaryotes. Okay, these are cells that are more complicated in general. They tend to be bigger in size, and they keep their genetic material in a specialized compartment called the nucleus. Okay, kind of like the nucleus of an atom. Okay, that's where the genetic material is going to be. That's where the DNA is going to be. And they have many organelles. Each of the organelles have a specialized function. Okay, so that's an organelle, that's an organelle. All these things are organelles. So each of them has a specialized function. And there are many different kinds of organelles. So human cells, animal cells, plant cells, these are all eukaryotic cells. Again, they are bigger in general. They are complex. They have lots of organelles, and they keep their genetic material inside a nucleus. On the other hand, the opposite of eukaryotic cells are prokaryotic cells. Prokaryotic cells are also known as eukaryotes. Okay, they are typically smaller, simpler in structure. They do not have a nucleus. So their genetic material just kind of flows around in the cytoplasm. It's not restricted within the nucleus. And they have fewer organelles. Bacteria are prokaryotes. Okay, so animal, plant cells, eukaryotes. Bacteria cell, prokaryotes. Okay. You don't have to memorize these labels, okay? Like it's not important at this moment. All right. So one of the three common features of a cell is that they have a cell membrane, right? Another name for cell membrane, as I have been telling you, is plasma membrane. So this is not the first time you saw a picture like this. I also show it to you in lecture 2, right? The cell membrane is made up of these phospholipids, right? And you might remember for the phospholipid, there is the hydrophilic head as well as the hydrophobic tails, right? And you will have two layers of these things with the hydrophobic tails kind of sandwiched in between. Okay, remember I told you to think of a peanut butter sandwich? The bread is like the hydrophilic head, and then the peanut butter in the middle. That's like the hydrophobic tails, right? So together, this sandwich forms a selectively permeable barrier. What that means is it helps control control what goes in and out of a cell. After all, you need to have some regulation. You cannot let everything just pass through, at free will. You want to keep the dangerous stuff out of the cell, and you want to bring in nutrients, and you want to keep them in the cell. So the cell membrane is able to help regulate what goes in and what leaves the cell. That's what selectively permeable means. And it also helps to maintain an internal controlled environment, right? Part of homeostasis, as you've learned in lecture 1. Okay, so we're actually going to learn a great deal about the cell membrane in lecture 4. There's a lot more information related to, what's in it and so on and so forth. But for now, just knowing this is enough. So I already told you it is a phospholipid bilayer. Bilayer means 2 layers, right? 2 layers. 2 layers. And with the tail sandwiched between. This is an actual picture of the cell membrane using one of those expensive microscope. You can see the 2 black lines here. Right? The black lines, those are actually the hydrophilic heads. Okay, and the little gray scale looking thing in between, the little gap between the 2 black lines, that's where the tails are. Okay, this is the outside of a cell, and this is the inside. Okay. I will only give you cartoon pictures to identify on a quiz. Okay, you're not responsible for these real pictures. But I'm just showing you real pictures so that you know, like, these are real, right? It's not like makeup stuff, right? So it turns out some of the organelles, some of the mini structures that are inside the cell, they also have their own plasma membrane. Okay, so if you think of a cell, that's the cell membrane, that's a plasma membrane on the surface. But then, as you will see, there are some other organelles inside. They would have an additional layer of plasma membrane. Okay. And that is it for the moment. Does anybody have any question before we maybe take an early break, and then we're gonna do some drawing when we return? Okay, so we will resume at 1:35. And then please go get some colors. Okay? You're gonna need some colors and markers and whatnot when we return. Okay, see you in a bit. So I want to take the next 20 minutes maybe to draw something together. It will help you understand it a little bit better instead of just watching it off the PowerPoints. So here we go. Please get a piece of paper and draw along. Okay. So this is I would recommend getting like a piece of paper and doing it sideways. Okay, I'm gonna start here and I'm gonna move down and maybe a little bit to the side as well. All right. So let's start with a cell here. Okay, so there is a cell. This is a eukaryotic cell. Eukaryotic cell. And mind you, they are bigger. They are more complex. They have a nucleus. That's the key difference between the eukaryotic cells and the prokaryotic cells, right? So that's the nucleus. And I want to take this out. I want to take the nucleus out and draw it a little bit bigger. Now, I recommend you draw it as best as you can right now, and then just try to reorganize your notes later on after the class. Okay, so here is the nucleus. And I hope you have colors with you. The nucleus is actually surrounded by an envelope. Okay, let me see if I can do this thing properly. There we go. You can probably do a little bit better than me on your paper. Okay, so there is an envelope here that surrounds the nucleus. Okay, that is called the nuclear envelope. And it is a membrane that controls movement of substances into and out of the nucleus. Okay. Now this nuclear envelope has some gaps in it. It has some openings in it. Okay? So like, you know, you can try to do that on yours. Okay, so these little openings, they are called nuclear pores. Okay, nuclear pore, and then another pore here and another pore, right? So the pores everywhere. So if things want to go inside or out of the nucleus, they're gonna have to go through the pores. Right? So things can pass through the pores and go into the nucleus. Similarly, they could leave the nucleus by squeezing through the pores. Okay. Just stop me if I'm going too fast or if you don't understand what I'm saying. Now, inside the nucleus is an other circular structure. Okay, so like this whole thing is a nucleus, right? And then inside the nucleus is an other thing, which is what this circle is. That's called the nucleolus. The nucleolus. Now, just to put things into perspective, in case you get confused, this is the cell membrane. Okay, that's the cell membrane. Okay, and then all this stuff here is the cytoplasm, right? I'm just zooming in into here. Do you understand that? Okay, we're just trying to magnify. Okay, so let's come back here for a second. I will, of course, post this online afterwards. So never mind what the nucleolus do for the time being. It's just something that's on the inside. Now, this space here, right, inside the nucleus, but outside of the nucleolus, that is where you are going to find genetic material. Okay, so these are genetic material. Okay. These are called chromatin. Okay, chromatin, which is basically DNA packaged with protein. Okay. DNA package with protein. I will show you a picture of what that means in a second. But just draw whatever you can first. I hope you're using color. If everything is black and white, it's super confusing. You know, it might not make sense later on when you look back at it.
It means that it is chromatin is outside of nucleolus.
That's right. The chromatin
Right.
is inside the nucleus, but outside of the nucleolus.
Excuse me, Professor, I have a question. This is the, you said, eukaryotic cell. But this
The
is different with atom? This is
What is the difference of what? I'm sorry, I missed that
for
part.
example, as you, Professor, you touched last week about atom, nucleus. So this is
Oh,
different.
yes, no, yeah. So atom is like the carbon, the hydrogen, the oxygen, right?
This
So
is different.
now we're all the way at the whole cell, the whole cell. So the cell can be eukaryotic cell, which is like us, animal cells and plant cells.
But it has nucleus, the same atom.
Yes, it's the same name. Yeah, nucleus just means a core. Okay, so I know. Oh, I see where the confusion is. Yes, there is also nucleus in an atom, but that is not the same as the nucleus we're talking about here. This is the
Okay,
nucleus of a cell. Okay.
I mixed. Okay, thank you.
Okay, so I want you to imagine this, guys. Okay, again, you don't have to draw this necessarily. You can just try to this next part don't draw. Okay, just like try to visualize it. Okay, think of an avocado. Okay, can you think of an avocado? Okay, this is an avocado, huh? Don't draw this. Okay, I have a lot more important things to show you afterwards. Okay, but this is an avocado. Right? Okay, so that's the avocado. Okay. The seed in the middle, it's like the nucleolus. Okay, I'm trying to give you like a visual ways to relate, right? This is just an analogy, right? Okay. So the seed is like the nucleolus. The skin is like the skin is like the what? Help me out.
Nuclear envelope?
Yeah, it's like the envelope. Very good. Okay, and the meat of the avocado, that's like where you will find the genetic material, okay? If the avocado is a nucleus. All right. Now take a toothpick, and you poke a bunch of holes on the skin. Poke some holes, right? These holes would be like the nuclear pores. Can you visualize that? It's hard to see on a cellular level, right? But like, think of objects that you are familiar with and you compare the analogy. Okay? All right, I'm gonna take the avocado away. At least maybe make it smaller. We don't need it anymore. You know what? Let's just erase it. We don't need it. Okay, so let's go back to the PowerPoint briefly. This here explain what chromatin is. Okay? We already know about DNA, right? DNA is the long double helix we learned last class, but it's very long and it gets tangled up easily, and think of like sewing threads. Okay? How do you pack the sewing thread nicely into a case? Well, you need to wrap them around the spool, right? That's what we are doing here. The DNA, if you just try to shove all of them into the nucleus, they're going to get tangled up. They're going to get broken up. We don't want them to be all unorganized. So we wrap them around packaging proteins, which are what these yellow things are. And eventually they become what's called chromatin. Okay, now you might have also heard of the word chromosome before, right? Chromosome. And on TV and movies, you might see them represented by these X's, right? So chromosome, it's essentially same as chromatin, but it's even more compact, more condensed, right? So regularly, when a cell is not dividing, the genetic material is in the form of chromatin. It's only when they're trying to divide, when they try to split, then the chromatin becomes the chromosome. Okay? But don't worry too much about it for now. Just know that the chromatin is essentially DNA packaged around proteins. Why do we need to package them? Because there is too much. There's too much DNA. We cannot fit them. So we want to, condense it a little bit. So that's what chromatin is. Okay. Now we will continue our drawing. We'll continue our drawing here. It turns out that the nuclear envelope is not going to stop around the nucleus. Okay, the nuclear envelope will actually continue to expand outward into the cytoplasm. Okay, and it will form these, networks like this. Okay, and then there are even more networks further out. Okay. And some of the networks are gonna have these little dots on them. Okay. Some of them are gonna have these little dots on them. And others will not have the dots on them. Okay. So let's talk about these little dots first. These little dots, they are called ribosomes. Okay. And ribosomes, they help make protein. They are important for protein synthesis. Okay. You've already learned that to make proteins, we need to connect amino acids together. How does the cell do that? Well, it relies on the ribosome to do it. The ribosome will connect the appropriate amino acids together and help build the protein. Some ribosomes will be stuck to this purple network. Other ribosomes will be floating around in the cytoplasm on their own. Okay, so these are also ribosomes, but these are free floating.
Professor, sorry, I didn't get. So what the network come from is
Okay,
from
so
the
you understand the envelope, right? The purple thing that goes around the nucleus.
Yes.
So that envelope extend outward from the nucleus, and then it forms this purple network, tunnels. Okay.
Tunnels, yeah.
And then further out, you also have like more tunnels, which is also an extension of this network. The difference between the purple and the pink is that the purple have these ribosomes on them to help them make proteins.
Thank you.
But not the pink. Okay? It's a lot of information, but we're just trying to give an overview right now. And once we've gone through the PowerPoints, hopefully it will make more sense. Okay, and that's part of the reason why I want you to draw it out, so you have it next to you when we go through the slides, and you can match the parts together. Okay, just one more thing on this picture, and then we're going to go back to the PowerPoints, but keep this with you. We're going to add more details to it as we progress. Okay, so back to the nucleolus for a second. Like, what does it do? Well, it turns out the nucleolus is the place where you make the ribosomes. Okay, so the nucleolus is the site. Okay, we'll just use simple English. Okay, this is where new ribosomes are made. Okay, so once you make the ribosomes, they can leave the nucleus through the pores. Some will be floating around on their own in the cytoplasm. Others will attach onto that purple network, which I have not tell you the name yet. That comes later. So any additional questions about this picture? Feel free to ask, please. Okay. Let's go back to the PowerPoints for a second. Okay, so basically this is a better picture, but it contains the same information. Okay, so let's just go through it one more time together. Hopefully drawing it out help you prepare for all the complexity here. This whole thing, that's the nucleus, right? And then the inside here, that's the nucleolus. You see all these little openings? What are they? I mean, the name is not really on the picture, but do you know what they are?
No
The
clue.
nuclear core.
The pores, right? The nuclear pores. Right? So if things want to get in and out of the nucleus, they have to go through these pores. Okay. Now, the chromatin is going to be the little fibers here, which it's difficult to see, but I just draw some more for you. That's the DNA packaged around proteins. Huh? Now, this nucleus is surrounded by the envelope, and the envelope, as you can see, extend out into the cytoplasm, right? So we have some that are covered by the yellow balls and some that are not. Okay? The yellow circles, they are the ribosomes, right? The ribosomes. Do you have a question, Fatima? No, that's okay.
I'm sorry.
No worries, no worries. Okay, so moving on. We talked about the chromatin already, right? And now this talks about the nucleolus, the thing that's inside the nucleus. And that is important for making the ribosomes. Okay, it's important for making the ribosomes. The yellow dots, right? The yellow dots in the diagram. Okay, so like I told you, some will be free floating in the cytoplasm. Others will be stuck to a network, which I haven't tell you what is called yet. Okay, like this purple network right here. They have the ribosome stuck on them, right? But it doesn't matter whether they are floating on their own or they're stuck to the network. The function of the ribosome is to make proteins. All right? Protein synthesis, connecting the amino acids together. if we take a closer look at the ribosomes, it's actually made up of 2 subunits. It's like a burger, right? The top patty and the bottom patty, right? The large subunit and the small subunit. They are usually apart, but when they have to make proteins, they come together. The ribosome is, even though its job is to make protein, but itself is also made up of protein and a little bit of RNA. Okay. These are the 2 ingredients that makes up the ribosome. And where does ribosome come from? The nucleolus, right? The nucleolus. So all these little black dots here, those are the ribosomes. And you can see they are kind of everywhere, right? They're everywhere. So, as I already told you several times, the ribosomes are made in the nucleolus and exit into the cytoplasm through the nucleopores. While some are freely floating in the cytoplasm, others will be attached to a network. Okay, so that network has a name. Right? It's called the endoplasmic reticulum. We will come back to that in a bit. Okay, don't worry about writing it down for now. Okay, just know that some are floating, the ribosomes, others are attached to this network. Any questions? You guys are very quiet. I'm not sure if you're with me.
We are trying.
I know it's a lot of information. Any questions though from anyone?
Yeah, I have to make sure one question, Professor.
Mhm.
So you told us before chromatin is like what I'm thinking about. I have some idea about chromosome, and chromatin is the planty or the planty number of chromosome. Is that
Okay,
correct?
so DNA is just on its own the genetic material, right?
Yeah.
Think of you know, sewing thread. I keep on saying. Let me see if I can search up a picture. Maybe it will help you guys understand a little bit more. Give me a second, okay? All right, this is gonna work. It's coming, okay? So so look, this is sewing thread, right? The individual thread, that's like the DNA. We have 46, 46 DNA, right, in the body. We have 46 different type of DNA. And you can imagine if you have all these sewing threads that is not wrapped around a spool is like the core, right? Then they're gonna get tangled up. You agree?
Yes.
So when you wrap the DNA around the core, around the protein, like this is essentially what we call a chromatin. Okay, it's nicely packed with the protein and it's, you know, very organized. They're not going to get tangled up. That's chromatin. Okay.
Okay.
Now, as organized as this is, if I want to move the chromatin around, they could still come loose. Okay, they could still get tangled up with something else. So when the cell needs to move their genetic material, and they need to do that when they're dividing, for example, then you take the chromatin and then you compact it even more. Okay, you compact them even more. You coil them up even more. You package them even tighter. So the only difference between chromatin and chromosome is how compact it is. Okay?
Yeah, I understand.
That's all.
Yeah.
Any other question? All right. So back to this situation, right? I have one more concept to show you. This is called the central dogma of biology. Okay? This means like central idea, central theme of biology. Okay? And we're going to revisit this process again and again throughout the course. We have DNA in the nucleus. Okay? And what information is in the DNA? Well, you might remember DNA contains instruction to make what? We learned this last week. What kind of instruction does DNA contain? That's okay. It contains instruction to make protein. Okay. It has the recipe for the protein, if you will. Right? But we don't want to take the DNA.
Vincent, I have a question. So
Yeah.
when you say to make protein, is it like the food we consume, like after it's been broken out to amino acids, or what protein is DNA making?
Yeah, okay. So like an example of a protein that we learned last time is like insulin, right? Do you remember insulin?
Yes, yes.
Okay, so we make insulin, we need it to, help control our blood sugar, right?
Sugar, yeah,
So,
right.
so to make insulin, we need to connect the right amino acids together.
Okay.
But which amino acid do we connect together? Well, all that information is in the DNA.
Oh, right. Okay.
So any kind of protein that you need to make an enzyme, the DNA is going to tell you how to do that. Okay? But the DNA is really big and really long, and we don't need to have all the instructions. Think of like a cookbook, right? If you want to make some kind of pasta for dinner, you don't need the whole cookbook, right? You only need the page that tells you how to make the pasta, right? So in the same way, if we want to make insulin, we don't need the whole DNA. We only need the section of the DNA that tells us how to make the insulin. So what we do is we copy the section that we need. Okay? So again, think of having this cookbook from the local library. They don't let you sign it out of the library because it's the only one they have or something, okay? But you really want that recipe, what are you gonna do? You photocopy, right? The recipe, right? I guess nowadays you can take a picture with your phone, but it's the same logic. You're not gonna take a picture of the whole book. You only take a picture of the pages that you need, right? So we only make a copy of the DNA for the proteins that we want to make. And that process is called transcription. Transcription. So a copy of DNA is made in the form of RNA. So that RNA, which now contains the instruction to make the protein that you want, is going to exit the nucleus through the pores. And then when it's in the cytoplasm, the ribosome, which is the blue thing here, is going to decode the instruction and then actually make the protein. So you see these circles in the protein, that's the amino acid. The ribosome will read the instruction, the mRNA, and then actually make the protein. And that process is called translation. Okay? So when DNA is copied into RNA, we call that transcription. And then when the instruction is used to make the protein, we call that translation. Does anybody have any question?
Can you explain this dogma thing again, please? I didn't get it.
This transcription translation situation?
Yes.
Okay. Do you understand the instruction to make a protein is
Yes,
in
I
the
do.
DNA?
Yeah.
You understand what that means, right? Okay.
Mhm.
So the DNA is really big, right? It has instruction to make all the proteins in the entire body. And we have like a lot of proteins. We're talking about thousands of protein, right? Okay. So at any given moment, you do not need to make the thousands of proteins. You only need to make the one that you need, right? That's why we are only copying the section of the DNA that you need in the form of RNA. Okay, so that process is called transcription. Once you make the RNA, you can take that copy and give it to the ribosome. Right? Remember, the ribosomes are the ones that actually make the protein, right? So to get the instruction to do it, it will have to decode the RNA. And the process of doing that is called translation. Is that a little bit better?
Yes, it is. Yes, yes.
Right.
Thank you.
All right. So that is that. Okay, let's do some polls. Okay. Well, prokaryotes are bacterial cell, right? Bacteria cell. Okay, e.g. bacteria. And there's only one bacteria in the list. That is E. coli. Okay. Liver cells are human cells. That is, eukaryote. HeLa cell is also human, even though it's cancerous, but it's still human. So that's eukaryote. Amoeba is not a bacteria. I told you they live in pond water, right? So that is also eukaryote. Okay? The only one that's prokaryote is the E. coli. You probably have heard of E. coli before, right? It's causes contamination in things and can make people feel very sick if it's in your food. Okay, how about this next one? Okay, thank you to those who have participated. This is talking about the function of the nucleolus, which is to produce the ribosomes. Okay, this happens in the nucleus. It doesn't happen in the nucleolus. All right. Let me see if we should continue. I think, you know what, I think this is a good spot to stop. I mean, we don't have a lot of time. We only have like about 5 minutes, and it's already a lot of information. So I'll stop here. We'll continue with our drawing next time. We still have a lot of things to add to this this picture. So good luck with preparing for the quiz. Remember, we have class on Wednesday. Okay, so do plan to attend. Okay, plan to attend. And you can take the quiz either in the morning or all the way up to 11:59 p.m. So I'll stick around for a few minutes in case you have questions. Otherwise, you're free to go. Okay, see you all on Wednesday.